The cell cycle is a series of events that take place when a cell is growing and dividing. We can represent it as a circle representing time, and the cell is going to go through these phases from the time it is, quote, born until it divides to produce two daughter cells. These daughter cells can then go through the process themselves. Okay, the cell cycle is a series of events, and we can categorize these events into phases. We can divide the cell cycle itself into two phases, interphase and M-phase. Interphase includes the time when the cell is not actively dividing, and M-phase, sometimes called the mitotic phase, is when the cell divides into two cells. We can further divide interphase into three distinct phases, G1, S, and G2. Let's walk through the cell cycle and briefly describe what happens during each phase. G1 is the first phase for a new cell. G stands for gap because when dividing cells were first observed under a microscope, they didn't really seem to be doing anything. We know now that actually the cell is quite active with preparations to divide. The cell is making the materials it will need to divide again, including energy reserves in the nucleotides and proteins needed to copy or replicate its DNA. During G1, the cell also has a, quote, choice. Now, of course, cells aren't conscious and they aren't really making choices, but essentially there is a point during G1 when if the cell receives a signal from the outside, or if it doesn't have enough resources to keep moving through the cell cycle, it will actually exit the cell cycle and enter a phase called G0. G0 is outside of the cell cycle. Some cells enter G0 temporarily and then re-enter the cell cycle where they left off. Other cells will never re-enter the cell cycle. Now they are perhaps very active in other ways, metabolizing and performing whatever functions they're specialized, but they're no longer going to divide, so they remain in G0 permanently. Examples of these cells include neurons and cardiac muscle cells. Very active cells, of course, with a very distinct function but no longer dividing. Sometimes we call these cells post-mitotic because they're not going to undergo mitosis again. Alright, but let's assume that our hypothetical cell is going to divide and keep progressing through the cell cycle. After G1 phase, the cell will enter S phase. S phase is very important because this is when DNA is replicated. In other words, all of the chromosomes of the cell are going to be duplicated. That's represented here, but do keep in mind that the chromosomes don't actually look like this at this point. They're still relaxed and decondensed in the chromatin configuration. They won't look like this until mitosis during M phase, but it's impossible to really see the duplication well in that configuration of chromatin. So we have our duplicated chromosomes, and they consist now of two identical molecules of DNA, which we now call sister chromatids. The sister chromatids are held together at a location called the centromere. Later, in cell division, the centromere will be an important place because it is where a protein complex called the kinetochore will assemble, and the kinetochore will be the place that the microtubules attach to pull the sister chromatids apart. After S phase, the cell will enter G2 phase, which is another gap phase or growth phase in which the cell makes final preparations to divide. We have duplication of some of the cell organelles, and the cytoskeleton is going to begin being remodeled for cell division. Okay, the cytoskeleton. This is a network of protein filaments that are present in cells. The protein fibers come in different varieties. From smallest to largest, they are actin filaments or microfilaments, intermediate filaments, and microtubules, which are made up of tubulin monomers. So all these are made up of proteins. And the cytoskeleton has many functions. It helps to give the cell shape and structure. It can interact with fibers outside the cell, such as connective tissue, and it's involved in movement. When I say movement, we could be talking about a couple of different kinds of movement. First, in some cells, it facilitates movement and migration of the whole cell. For example, when a single-celled organism like an amoeba moves, it is relying on the cytoskeleton. The protein fibers can get longer in the direction that the cell wants to move, and they can get shorter at the other end of the cell. This video is one I took in the lab, and I've sped it up 4 times so you can see the movement a little bit better. Similarly, if we look at the tips of baby neurons, which are extending and trying to find another cell to connect and form a synapse with, they are relying on the cytoskeleton to facilitate this movement as well. Aren't they adorable? But the cytoskeleton is also really important for movement inside the cell. Organelles and vesicles can move along the protein fibers from one place in the cell to another. And in cell division, the cytoskeleton is going to help move the chromosomes. What you're seeing here is mitosis, On the left are microtubules that are labeled, and on the right you're seeing just the overview of the cell. Now we move into M phase, which is when the cell actually divides. We'll dive into the phases of mitosis a little later, but briefly, the chromosomes condense, the microtubules attach to protein complexes called the kinetochores that assemble at the centromeres, the sister chromatids are pulled apart to either side of the cell, and finally, the cell splits into two daughter cells. Each of these two daughter cells can now go on to G1 and begin the cell cycle anew.